Okay, the next two chapters are talking about muscular tissue, and that's chapter 9, and then chapter 10 is going to talk more about muscles as individual organs. Now, the special characteristics of muscle tissue, there's 4 things that muscle can do. First of all, it's excitable. That means it's responsive or irritable. It's able to receive and respond to a stimuli. It's contractible. It's called contractility. It's able to shorten forcibly when stimulated. And it's also extensible. It's able to be stretched, and then it's elastic. It's able to recoil to its resting length after it's been contracted. So when you look at types of skeletal muscle, this chapter primarily focuses on skeletal muscle, but there's also, remember, smooth and cardiac muscle. So this chapter mainly focuses on how skeletal muscle works, and we're going to get into some physiology in this chapter as well. So here's some characteristics of skeletal muscle themselves. They are individual organs. There's over 600 of these in the body, and they attach to bones and to the skin. Elongated cells, which are called fibers. These cells can be as long as over a foot in length. They're very, very long, so that's why they're called fibers. They have the striated or striped appearance. They're voluntary. You control them. They contract very rapidly, they tire easily, and they can be very, very powerful. Their forces can range from a fraction of an ounce to over 70 pounds. And they do require nervous system stimulation. If you want a muscle fiber to contract, you have to have a nerve fiber attached in some way. Now this picture here is looking at a piece of a muscle fiber, and you can see that it has a covering. One of the things about muscle cells is some of the microscopic structures have their own little names. So the sarcolemma is the name for the cell membrane. That's right here. So sarco is one of the terms you'll see associated with microscopic structures. So sarcolemma. And then you see that the nuclei generally are outside of this region that's filled with all these protein fibers that are called myofibrils. This is a myofibril kind of blown up so you can see it in more detail. But skeletal muscles are filled with these protein bundles that are called myofibrils, and this is a piece of a muscle fiber. There is a little covering that goes around the cell membrane, the sarcolemma, and that's called the endomysium. And when we get to chapter 10, we'll talk about the organizational plan of muscles, how cells are grouped together, and then larger structures actually form the muscle. So a muscle fiber is an elongated multinucleate cell. It has a banded, striated appearance. Now, the myofibrils are rod-like contractile elements. And these myofibrils are made up of subunits that are called the sarcomere. Now, I want you to know that the sarcomere is the structural and functional unit of muscle fibers. Okay, that's where the action is going to occur. Now, these myofibrils, if you read this, are rod-like contractile elements that occupy most of the muscle cell volume. Composed of sarcomeres arranged end to end, they appear banded, and bands of adjacent myofibrils are aligned. So this arrangement, we're going to find out, contributes to the banding pattern that you see in muscles. Now, this is looking at a sarcomere. So this is a sarcomere, and you see there's red fibers here, and then you see blue fibers, and then you see these little yellow fibers here. Now, the red ones are called myosin, and the blue ones are called actin. And because these are thicker, they call the myosin the thick filament, and they call the actin the thin filament. And see how they overlap? So this little arrangement here is what we call a sarcomere. And what's going to happen is when the muscle fibers contract, these little pieces right here, the head of the myosin molecule, they're going to reach up and grab onto this blue one, and these reach up here, these reach up here, and they pull the blue filaments closer together in the middle. And I'll tell you, a really good way to visualize this is if you go on to YouTube and look up videos that have the theory of muscle contraction. It'll show you pictures that are moving pictures showing you this in action. So I would recommend that you do that and just get a better look at how this is actually occurring. So contractile filaments are of two types, thick and thin. Remember, the thick is the myosin, the thin is the actin. So this banding pattern, wherever you just have myosin, that is called the dark band or the A band. This is important for you to know too. So wherever the myosin is, that's the dark. And in between myosin bands, where you just have actin, that's called the light band. Now take a look at the words. Dark has A in it, so the dark band is the A band. Light has I in it, so that's the I band. And here you can see again structures we already talked about. The myofibril, there's mitochondrion in these muscle fibers to make ATP. There's your cell membrane, the sarcolemma, and here's your nuclei underneath the sarcolemma. So here you go. A band, myosin, those red filaments appear dark. In between, adjacent, there's another A band right there, the next one next door. There's where you just have the blue filaments. That's the I band. That's light. So these are what this arrangement of the sarcomeres is what makes the banding striated pattern that you see in skeletal muscle. It makes it look like a candy cane. A band, myosin. I band, no myosin, just actin. And the sarcomere itself goes from the Z disc to the Z disc. Okay, that's the length of the sarcomere. Z disc to C disc. In the middle, there's these bands that run perpendicular, and those are called the M line. And the separation point between two adjacent actin filaments is called the H zone. Here's another picture showing you those M-line filaments that run perpendicular to the other filaments. And there's the Z disc, there's the Z disc, that's the length of the sarcomere. Here's your myosin filaments, the blue are the actin. Remember, myosin is thick, actin is thin. And then the myosin is anchored to the Z disc by these little, they look like springs, don't they? Those are elastic filaments. So when the sarcomere is contracted, the springs are compressed. And I'll show you a picture of that in a minute. So this is what accounts for the recoil action of the muscle fiber. When these springs are compressed, they want to recoil back to the resting length and bring the muscle back to its original shape. So here's another picture. It's showing you the actin, the myosin, and it's giving you more detail about what the myosin heads look like. And they kind of look like golf clubs. So it has a long tail, and it has this head piece that has an ATP binding site, and there's a flexible hinge because this thing is going to be able to bend. And then the top of it, it has actin binding sites. And then over here on actin, the blue filament, it looks like a series of beads connected together. And over top of what are called binding sites, these little holes here that you see, there is this filament that's called tropomyosin. Now, tropomyosin blocks the binding sites, and there's a lock that fits over this tropomyosin blockade that's called troponin. Now, troponin, when it's in the lock position, holds that tropomyosin blockade over the binding sites on actin. And what we're going to have to do is we're going to have to get something to move that tropomyosin blockade away so that the actin binding heads on the myosin can reach up and touch the actin binding sites and slowly but surely on this side and this side pull these actin filaments closer together in the middle, and that's going to shorten the sarcomere. This is showing you a fully relaxed sarcomere. This is an electron micrograph. See the space in between the actin filaments? And then here's a fully contracted sarcomere. Look, no space in between because they've been drawn closer together. And actually in this picture, you cannot see the next myosin band, the A band next door, but here you can, because this one here has contracted, okay? So just bringing, actually when contraction occurs, all you're doing is bringing the actin filaments closer together. Now this is what happens when a muscle cell is actually going to be stimulated to contract. This is an axon terminal of a nerve fiber. And when a nerve fiber is stimulated, an action potential goes down the axon of that neuron to the end, or what they call the axon terminal. And in the axon terminal, there are these little vesicles that are filled with neurotransmitters. And in this case, the neurotransmitter is called acetylcholine. So these vesicles are here. Here's the mitochondrion, all right? So the action potential comes down. And when it does, it opens up voltage-gated calcium channels. Okay, so the channels open, and that calcium is important because it causes the acetylcholine that's found in the vesicles to be released. So ACH, capital A, capital C, little h, is the term for acetylcholine. Okay, that's the short abbreviation for acetylcholine. You can use that. It's a lot easier than writing out that long word. There's acetylcholine up there spelled out if you want to have that, for your reference. So acetylcholine is released. Remember, exocytosis is those little vesicles fusing with the cell membrane and releasing the neurotransmitter. And then the acetylcholine diffuses across the synaptic cleft. The synaptic cleft is actually the gap between the nerve cell membrane at the end here and the muscle cell membrane. So there's a little space here, and that's called the synaptic cleft. I would make sure you remember these terms as we're going along. The acetylcholine diffuses across that synaptic cleft, and it binds to receptors on the sarcolemma. Now, those receptors are called acetylcholine receptors. So acetylcholine is going to bind to those receptors and open them up. So when acetylcholine binding opens the ion channels, okay, what it does is it's going to cause an exchange between electrons. So what's going to happen initially, these channels are really called sodium channels, and it lets sodium into the membrane. And when that happens, that is going to do something called depolarize the membrane. Now, the effect of acetylcholine doesn't last a long time. It's terminated by its breakdown because there's this enzyme that's called acetylcholine esterase. And acetylcholine esterase, that's a very long word, right? That's an enzyme that breaks down acetylcholine and degrades it so that the action of acetylcholine is not maintained for a long period of time. You have to continually get nervous stimulation for this to have any type of activity. So what happens is you have more sodium outside the cell normally and very little inside. Inside the cell you have a lot more potassium, okay? And when these acetylcholine receptors are opened, you're going to let sodium in. So ordinarily the resting potential is very low inside the cell because you don't have that much sodium inside. So what happens is when you open up the channels, the sodium comes in. And when that comes in, sodium has a plus charge. It makes the inside of the cell less negative. And then what happens afterwards is the channels close. And then potassium channels open. Now remember I said the potassium was in higher concentration inside the cell. So potassium goes out, and it has the same charge as sodium. So when potassium goes out, you repolarize the membrane. So you've let sodium in to depolarize it, and that's going to allow your action potential to pass through. And then you repolarize by letting potassium out. So let's just back up and review some of the steps that we talked about already. The action potential arrives at the axon terminal at the neuromuscular junction. This is the first phase. This is where the muscle cell is actually stimulated. Acetylcholine is released. It binds to receptors on the sarcolemma. Those are your acetylcholine receptors. Remember, sarcolemma is the cell membrane of the muscle. Ion permeability of sarcolemma changes because you're opening up these sodium channels. Local change in membrane voltage occurs, depolarization, because you're letting sodium in. Local depolarization in plate potential ignites an action potential in the sarcolemma. So by depolarization, you're able to pass that electrical signal from the nerve right through into the muscle fiber. The action potential crosses the entire sarcolemma. This is an electrical event. The action potential travels along these structures that are called T-tubules. And this is going into what's happening inside the muscle cell now. It's phase 2, called excitation-contraction coupling. And it goes to an organelle that's called the sarcoplasmic reticulum. Now, you remember us talking about the endoplasmic reticulum back when we were talking about cell parts, right? Well, in this case, that endoplasmic reticulum, which is called sarco, like some other sarco words we ran into already, the sarcoplasmic reticulum releases calcium. That's what it does in muscle cells. That particular organelle stores calcium. So this action potential traveling down the T-tubules, and the T-tubules, I'll show you a picture in a minute, the sarcoplasmic reticulum is on either side, and that electrical event causes the sarcoplasmic reticulum to release calcium. The calcium binds to troponin, and remember, troponin is the lock that's holding that tropomyosin blockade over the actin binding sites. So you might have to go back over this a couple times to review what I talked about and see the pictures. But calcium binds to troponin. That's the key. Calcium is the key that unlocks the lock. And now the tropomyosin blockade moves away from the actin binding sites. So the myosin binding sites are exposed on actin, and the myosin heads bind to actin, and contraction can begin. So I want you to remember, it's all due to this depolarization wave, bringing in sodium. And then you're releasing calcium from the sarcoplasmic reticulum. And by doing that, then you're able to get that calcium, which is the key, bind to troponin, unlock the lock, move the blockade away from the binding sites on actin so the myosin heads can bind. So here's a look at it in picture form. There's your axon terminal. There's the synaptic cleft, little vesicles filled with acetylcholine. Remember, your receptors are on this blue membrane. And once the acetylcholine is released, the action potential goes down. Here's your T-tubule. The T-tubule is actually in between adjacent areas here where you have sarcoplasmic reticulum, which is storing the calcium. And you can read this over yourself. I'm not going to go over all of it, but you can read it over. And so what's happening is when the calcium is released, it binds to that little yellow structure there. That's your troponin. And when troponin is unlocked, the tropomyosin blockade is moved away. And then you're going to see that your myosin head here can reach up, grab onto the binding site, and see how it pulls forward. And so it's pulling the actin filaments forward. And remember, we have actin filaments on either side of the sarcomere. So on both sides, this is occurring and bringing those actin filaments closer to the middle. And after the contraction phase is over, then relaxation occurs and calcium is actively transported back into the sarcoplasmic reticulum. And when calcium is removed from the troponin, it returns to its original locked position and puts the blockade of tropomyosin back over the actin binding sites. And then this ends the muscle contraction phase. Now this is what happens with myosin. Because remember, the myosin heads are the ones that actually move. And what makes the heads move is ATP. You have to have ATP. So in the first one, you can see here you have ADP and phosphate. That's when the ATP is actually broken apart into two pieces. So when the myosin reaches up and grabs an actin binding site, that forms a cross bridge. And the energized myosin head is energized because of the hydrolysis of the splitting of ATP. And when that happens, the energy that's stored in the ATP molecule is transferred to the myosin head that can produce the action. So the energized myosin head attaches to an actin myofilament, forming a cross bridge. And then the power stroke, or the working stroke, is when the head pivots forward and pulls the actin filament closer to the middle of the sarcomere. And your ADP and phosphate are released. Because remember, think about proteins. When something happens, when they move or something binds and changes shape, then that's when you have something usually occurring. In this case, binding of ATP causes a change in shape. Releasing the ATP in the phosphate causes a change in shape. The swiveling motion, okay, all tied into proteins and how they operate. So by releasing the ADP and phosphate, the myosin head pivots and bends, changing to its bent. This is called the low energy state. And as a result, it pulls the actin filament towards the M line. Now, I always think about this as maybe something similar to a little cap pistol that we had as kids. And when you pull the hammer back by using the energy from your thumb, you're pulling the hammer back. Then you pull the trigger, and when the hammer goes forward, then the hammer's in the low energy state. You have to pull it back again to put it in the high energy state so that you can pull the trigger and make the gun fire. So this is called the low energy state after the actin filament is pulled forward. Now, in order to break the cross bridge, you have to bind ATP. So by binding ATP, that changes the shape of the myosin head so that it pulls back away from the actin filament. So that breaks the cross bridge. So ATP has to bind in order to break that cross bridge. And then by ATP hydrolysis, that transfers the energy from ATP to the myosin head. That's called cocking of the myosin head. So as ATP is hydrolyzed to ADP and phosphate, the myosin head returns to its pre-stroke high energy or cocked position like the cap pistol, pulling back the hammer and ready to fire. And you can see it goes around and around and around. So energized head attaches, pull forward, ATP binds, pull back, hydrolysis, push forward. That's the idea behind how the myosin head actually operates. And all this requires ATP. So rigor mortis, what happens with rigor mortis? Cross-bridge detachment requires ATP. So if there is no ATP, over time, the cells start to lose their ability to produce it as oxygen levels go down. And within 3 to 4 hours after death, muscles begin to stiffen with rigidity at 12 hours post-mortem. The dying cells taking calcium leads to cross-bridge formation and no ATP generated to break the cross bridges. So because there's no ATP, the cross bridges stay attached and the muscles kind of freeze in place. And after a certain period of time, then muscle and protein decomposition reverses the process and rigor mortis will reverse itself. So review principles of muscle mechanics. Let's look at different types of contraction that may occur. Now, we do have types of contraction that may not shorten the muscle, and this is called isometric contraction. In isometric contraction, the muscle exerts a lot of tension, but it can't overcome the load. And this happens if you have a weight that's too heavy. Like if you go to the gym and the barbell is set to an excessive amount of weight, you can generate a lot of force, you can work up a sweat, but you'll never move the load. So that's isometric contraction. Yes, you're exerting energy, but you don't move the load. Isotonic, now the load's light enough. The muscle can shorten because the muscle tension exceeds the resistance or the load placed upon it. The muscle will shorten and the load is lifted. Force and duration of contraction vary in response to stimuli of different frequencies and intensities, because remember, with muscles you can generate different types of intensities based on the amount of muscle fibers that you recruit in when you're performing an action. Because I can flail my arms around and not use many muscle fibers at all, or I could pick up a weight, or I could pick up a table, and I'd have to recruit in, so I get different intensities of muscle contraction. So isotonic contractions, where you actually move the load, the thin filaments slide, and you shorten the sarcomere. There's two types of isotonic contractions, concentric and eccentric. Now, concentric contractions, the muscle actually shortens and it does work, moves the load. Eccentric contractions, the muscle generates force as it lengthens. Like if you are picking up a barbell, instead of just dropping it to the ground and slamming it to the floor, you can gently lay the weight down by lengthening the muscles and gradually releasing it. So that's what eccentric contraction is. You're generating force, holding the weight as the muscle's lengthening and gently letting it to the floor. So a motor unit. Well, then what's a motor unit? Because we said that muscles have different degrees of force that can be exerted depending upon the motor units that are recruited in. Now, a muscle as an organ, you can see it here, has many, many muscle fibers, and there they are, different muscle fibers. And you have this motor neuron that goes down into the muscle. Like say we're talking about the bicep brachii, the major muscle of forearm contraction. It's on the anterior surface of the upper arm. So if I wanted to contract that muscle and pick up a barbell in my hand and flex my forearm, what I'm going to do is I can recruit in one. If I don't want to have that much force, these motor units are all associated with various muscle fibers that are spread throughout the entire muscle. There's motor unit 1, and you can see it in purple here, and it's attached to that one, that fiber. And then this one here, motor neuron 2, is attached to this fiber, and this fiber. Now, if I didn't want to exert much force, I may just recruit in motor unit number 2. And so I'd get very light contraction of that muscle. I could pick up a can of soda maybe. But if I wanted to, say, pick up something heavier, like a 10 or 15 pound weight, I might need more. And it's not just 1 or 2, there's many motor units. So you can get quite a variation in the amount of force depending upon how many motor units you recruit in. So the definition of a motor unit, it's a branch of a motor neuron and all the muscle fibers it innervates. Now this is a diagram showing you the typical muscle twitch. And a muscle twitch has 3 phases. The first one is the latent period. The latent period is usually 1 to 2 milliseconds. You can see down here over time, this runs over 140 milliseconds. So what's happening during the latent period? Well, here's what's happening. The action potential is coming down the nerve fiber, gets down to the axon terminal, opens up calcium channels. The calcium channels make those little vesicles filled with acetylcholine, fuse with the axon terminal membrane. Acetylcholine is released by exocytosis, goes across the synaptic cleft, binds to acetylcholine receptors, opens up sodium channels. Then the action potential passes on to the sarcolemma of the muscle fiber down the T-tubules, opens up the calcium channels to let the calcium out of the sarcoplasmic reticulum, and then the calcium binds to troponin. Troponin moves the tropomyosin blockade away from the actin binding sites, and then myosin hydrolyzes ATP to go into the power stroke, attached to the actin filaments, and muscle will move. Well, all that stuff leading up to when the myosin binds and moves those actin filaments, that's all happening during this 1 to 2 milliseconds. It took me longer than that to say it, but that's like a review of what we already talked about. Then you get the contraction phase, okay? And these are thousands of seconds. So the contraction phase occurs, you get the tension, and then relaxation. A little bit longer time there. So those are the 3 phases. Now, depending upon the size of the muscle will determine the relative period of time that a muscle twitch takes to occur. The latent period is always the same, 1 to 2 milliseconds. But see this? This is an extraocular muscle. This is a muscle of the eye. Very quick, very quick. They always say the eye is faster than the hand. Well, yeah, sort of, look. And then the gastrocnemius is the superficial muscle on the calf, and then on the back of the calf. And this one is the one that's underneath, the soleus. So the gastrocnemius is pretty quick, running and jumping and changing directions quickly. And the soleus is kind of a slow aerobic muscle that's in for the long haul. So that one takes a lot longer to contract. Okay, so just the type of muscle it is, how big it is, what its function is in the body will determine what type of muscle twitch myogram you will get from that muscle. So contraction strength of twitches, they're stronger when stimuli are close together. So when you have back-to-back stimulation, you can get more and more contractions, and they're quicker to occur. And they vary with calcium concentration, because remember, calcium needs to be available in order to bind to troponin. And it depends on how stretched the muscle was before it is stimulated. Warm muscles contract more readily. When you warm up a muscle, it makes those enzymes work more efficiently, oxygen is released more readily, and those muscles work a lot better. And as muscles work, they get tired, because they start to build up lactic acid over time, and some people can go a long time and operate under aerobic conditions and not build up a lot of lactic acid, but over time, muscles will develop lactic acid as they fatigue. That's going to drop the pH. And remember what the pH does. A lower pH can denature proteins and interfere with the action of those enzymes. So it slows things down. And also hydration is really important for muscles to be able to work effectively. Now, if you have multiple back-to-back stimuli, the term recruitment is bringing in many motor units, and recruitment enables you to generate more force. Treppe is the staircase effect. What happens when you're warming up a muscle? That the muscle becomes more efficient, it generates more force. So if you're looking at a myogram, you'll see more tension generated as the muscle works up, starts to warm up, and it looks like a staircase. You get force, and the next one's a little bit more force, and a little bit more force, so it looks like a staircase effect. Wave summation, if you have a lot of contractions happening close together, it'll look like a wave. But you always have a latent period in between contractions, so that you never really have a straight line. It always looks like a wave because there's always a little bit of space in between. Even though it's not enough for you to visually see that, I can clench my fist and keep it closed, and it looks like there's no break in between, but actually there is, but not enough to really visibly see that. Complete tetanus is something like, if you did have that straight line, that would be painful. And its prolonged sustained contraction can be stimulated by exerting a lot of, or introducing a lot of high voltage, like if you grabbed a high tension wire. And with that type of tetanus, you can't let go, and it's extremely painful. That's why I said wave summation is normal, because you do have that little gap in between back-to-back stimulations for the latent period, 1 to 2 milliseconds. Here, you're overriding it by introducing a lot more voltage than what you normally would find in the body, and you don't really see that. So how do you generate ATP? Because we know we need ATP. We got to have ATP to be able to get those myosin heads to attach. So direct phosphorylation is a source of very little ATP production. And you do have something called creatinine phosphate in muscles. And creatinine phosphate can actually take a phosphate and transfer it to an ADP, which is a low energy form of ATP. And there's an enzyme called creatinine kinase that makes this happen, and you're going to get ATP. So it's transferred from the creatinine phosphate to ATP, and that gives you a very limited source of ATP productions. So you produce 1 ATP per creatinine phosphate molecule, and it provides about 15 seconds worth of energy, so very limited. So when you're exercising, short-term exercise, the first 6 seconds, ATP that's stored in muscle is used first. Then you have your creatine phosphate and ATP direct phosphorylation. And I'm not going to bother going through that. I'm not going to ask you anything about that. And then glycogen that's stored in muscles is broken down into glucose. And glucose is broken down inside the cytoplasm of the cell into 2-pyruvic acid, and that releases 2 ATPs. This is anaerobic, okay? So that's another source of energy that's very short, 30 to 40 seconds, and then the end of the exercise. So this is short-term anaerobic types of exercise. Now, if you want to exercise for a long period of time, what you need to do is produce the ATP, take it into the mitochondria, and completely break down the glucose, and this will give you a lot of ATP. Instead of 1 or 2, now you're going to get 36 for every glucose molecule. So you're talking about a lot of ATP, and you're not really exercising anaerobically, you're exercising aerobically. And you have enough oxygen to be able to fuel the process, and you can go for a long period of time. So when muscles fatigue, they are physiologically unable to contract despite continued stimulation. This occurs when ATP synthesis declines as glycogen is consumed. ATP shortage slows down the sodium-potassium ion pump. Now, you remembered us talking earlier about depolarization, repolarization, right? If you let sodium in and depolarize the membrane, your response is to let potassium out. But what that does is that's a reversal of the location of sodium and potassium. So to restore the membrane to the resting potential, you have to use the ATP-driven sodium-potassium ion pump. So as ATP is slowing down, you don't have this pump that pumps the sodium back to where it belongs and bring the potassium back into where it belongs before the muscle was contracted in the first place. And as that slows down, then sodium is inside the cell, and you're reducing the ability of having enough sodium outside to make the muscles contract by depolarization. So it's going to slow down the whole process. And lactic acid builds up because you're not able to create the ATP because of all the processes slowing down, and that lowers the pH, which is going to render a lot of your enzymes ineffective, and then that's going to contribute to functional fatigue. And fatigue of the central nervous system is not as well understood as the mechanics going on behind what's happening in the muscle, and that has a lot to do with a person's mindset. There are people that can override tiredness, fatigue, and will themselves to go a lot further than other people. There are some people, they run 10 steps and they say, well, I'm tired, I've had enough, I'm done. And there's a lot of people that can go run a couple miles and they say, you know what, yes, I'm tired, but I'm going to keep going. And that's all mental. So there's a lot, when it comes to exercise and endurance, a lot of it, yes, it's in your head. So post-exercise oxygen consumption, what you have to do to get the muscles back to the resting state. Okay, and this takes a lot of oxygen consumption post-exercise, beyond the exercise period. So here's the good thing about exercising, is your metabolism stays up for hours afterwards. You're burning more calories for a long time after the exercise period's over to put everything back to where it belongs before you exercised in the first place. And so this requires a lot of oxygen and a higher metabolic rate in the cells. So oxygen reserves have to be replenished. Lactic acid has to be converted back into pyruvic acid. Glycogen stores are replaced. ATP and creatinine phosphate reserves are replenished. And all of these things require extra oxygen, which occurs post-exercise. Okay, so the other thing is we have two types of fibers inside of the muscle. And it has to do with the speed of contraction. We have some that are called slow twitch fibers, and we have some that are called fast twitch fibers. And which is determined by the speed at which the myosin ATPases are able to split ATP, and the pattern of electrical activity of motor neurons. So here's what you need to know. Metabolic pathways for ATP synthesis. These oxidative fibers, these are your slow. They're slow twitch fibers. They're red. They have a lot of myoglobin. Myoglobin stores oxygen. And they're aerobic fibers. They're slow and therefore the long haul. Glycolytic fibers, these are your fast twitch. They use anaerobic glycolysis pathways. They're fast. They fatigue fast, okay, because they're anaerobic. And so they wear out pretty quickly. Those are called white fibers, okay? So that's the difference between the two. And actually, we're a blend of both, okay? We have a mixture. And you can train yourself to be more efficient at anaerobic types of exercise. You can train for marathons, long distance type of exercise, and make those oxidative fibers more efficient. So muscle strength and conditioning is determined by the relative size of fibers. Bulkier muscles, and if you increase the size of the cells through weight training, that's hypertrophy, you exert more force. The size of the motor units, how many muscle fibers are attached to each of those nerve fibers. The more muscle fibers you have attached, the more motor muscle fibers you have to contract, and that makes those motor units able to exert more force. As more muscle fibers are recruited, you generate more force. Frequency of stimulation. If you increase the frequency of stimulation over time, that allows for the transfer of tension to non-contractile components, and these non-contractile components are important for stimulating the muscle to generate more force. And these non-contractile elements are built within the muscle. They're called muscle spindles. And muscle spindles respond to muscle stretch. So as the muscle's being worked, it's kind of hard to explain, but as the muscle's being worked and the muscle's being activated over and over again, these non-contractile components will maintain that force. They kind of store the information to keep the muscle moving. And there's a length and tension relationship too. Muscles that are at 80% to 120% of normal resting length will generate more force. So this is looking at that length and tension relationship of the sarcomeres. Here you can see the sarcomere is at its optimum resting length. Here it's really contracted. There it's like really, really contracted and the fibers are overlapped. Here there's a big space in between the actin filaments, and here there's like really stretch or pull muscle. But you can see you get your greatest degree of contraction where you do have some overlap, not an excessive amount, but some overlap between the myosin and actin filaments. And less here because you don't have anything for the myosin filaments to really grab onto. And here it's already contracted. So that's what that means. Tension, length tension relationship of sarcomeres. And this is looking at the picture form of what we talked about earlier, and showing you that the number of muscle fibers recruited gives you more force, the size of the muscle fibers, high frequency of stimulation, back-to-back-to-back stimulations, and the muscle and sarcomere length being very important, that length-tension relationship. All this is involved in how much force, how many more cross bridges you can make and make that muscle contract. Resistance exercise is really important, and that's typically anaerobic. A lot of people do weight training. You know, that's really important to cause muscle hypertrophy, increase in size, and that's primarily due to an increase in muscle fiber size. Also, over time, you increase the number of mitochondria, which is going to cause you to produce more ATP, which is going to make the muscles more efficient. More myofilaments greater glycogen stores. Glycogen is our storage molecule for glucose, and the connective tissue in between the muscle fibers becomes more extensive and helps the muscle to be stronger and prevent injury. And so all this leads to increased muscle strength and size. So this is looking at the 3 different types of muscle that we know we have. Skeletal, we spend a lot of time on this. Cardiac, we know that cardiac is also striated, but this is not something you control. And in chapter 18 next semester, we're going to talk a lot about how cardiac muscle operates. And then smooth muscle, you see, is found in hollow organs like the stomach, the intestines, the bladder, blood vessels, but again, not in your control. And smooth muscle, I'm going to talk a little bit about how smooth muscle operates. Smooth muscle cells are kind of interwoven into a sheet. They're fat in the middle, they're tapered on the ends. And the way the nerve attaches to the smooth muscle is that it kind of spreads over the sheet of muscle, and it has little swellings in it called varicosities. And these varicosities have synaptic vesicles that store neurotransmitter. Okay, and it'd be acetylcholine, just like we had with skeletal muscle. But here's your varicosities. There's some mitochondria, and the varicosities release their neurotransmitters into a wide synaptic cleft. Okay, so you have a wide area that this is spread over. And the neurotransmitter is spread from cell to cell because muscle cells, smooth muscle cells, are connected by gap junctions. So those gap junctions enable the cells to work together as a group. So what happens is that you get the calcium coming in from the cytosol, and the difference with smooth muscle is that it has no troponin. It has something called calmodulin. And the calcium binds to calmodulin. So no troponin, remember that, because troponin is what you see in skeletal muscle. So it binds to calmodulin. Calmodulin activates this enzyme that's called a kinase. The kinase is what hydrolyzes ATP and passes this along to the myosin head. And then the myosin head attaches to the actin, and then you get the contraction of the muscle fiber. Now, smooth muscle, actually, because it operates like a sheet, it kind of contracts like a wave, and so it goes down the length of the muscle. So that's smooth muscle contraction. Muscular dystrophy is a group of muscle wasting diseases, and Duchenne's muscular dystrophy is the most common, and that's the one that they used to do a lot of telethons on TV for to raise money. It's the most common and severe type. It's inherited, it's sex-linked, so it's passed from mothers to son, carried by females, expressed in males, and actually 1 out of 3,500 live births will show muscular dystrophy, and it's as a lack of dystrophin. So dystrophin is something that makes the cell membrane of muscles more stable. And if you don't have this, what happens is it makes the sarcolemma very fragile. And so calcium will enter, and it damages the contractile fibers. The cells become inflamed, and muscle mass drops because the muscle will atrophy, but you do have a lot of inflammation. Can start as early as 2 or 3 years old, but the victims become very clumsy and they fall frequently. And every muscle usually is affected over time, and eventually the respiratory muscles are affected, and the victims die of respiratory failure usually in their 20s.